So prokaryote replication, it has various members that are going to assist in replicating the genome of the species in order to synthesize a newly replicated DNA. And so some of these players that we're going to be talking about include enzymes like helicases, primase, RNA polymerase, polymerase 1 and 3, and ligase. So the first step in DNA replication is the entire genome has to, or the DNA has to unwind. This is accomplished by helicase, an enzyme that splits the DNA down the replication fork and basically splits apart the double-stranded DNA to expose that single-stranded DNA. And the next player that comes into play is going to be primase. So primase is going to go ahead and synthesize that RNA primer that later on DNA polymerase is going to need to sit on that single-stranded DNA and start synthesizing new nucleotides and start replicating the genome. DNA polymerase 3 starts synthesizing the leading strand, and you'll learn that there are two strands, the leading and the lagging strand. The leading strand is actually a lot more simpler in our case because it doesn't involve as many glitches, but we'll go ahead and explain that later on. But basically, the DNA polymerase 3 sits on that RNA primer and it moves across the single-stranded DNA to synthesize a double strand, and eventually goes in the direction of 5' to 3' and is able to complete the synthesis of that strand. And there is actually another strand that is below it, you'll see, is referred to as the lagging strand. So this lagging strand is going to vary in the manner in which it replicates its DNA. So for the lagging strand, it is actually going to be discontinuous assembling of nucleotides, and this is because it is in the position of since it has to go from a direction of 5' to 3', it's going to be going the opposite direction as the leading strand, and since the replication fork isn't completely open yet, it's going to need to start synthesizing nucleotides while it is still opening up. And basically this occurs in a discontinuous manner. DNA primase once again comes into play to assemble that primer, which DNA polymerase 3 will then start to assemble nucleotide sequences. Since they are discontinuous, they're referred to as Okazaki fragments. And in the end, that primer is going to be removed by DNA polymerase 1 because our cells do not like have RNA in our genome, and RNA is going to be pulled out by that DNA polymerase 1. And finally, ligase is going to come into play to reattach or to ligature all those Okazaki fragments and to put it all together. So an interesting phenomenon takes place in DNA replication that is going to require the need of telomeres to solve an important problem that can result in many eukaryotes. And so these telomeres, basically for the leading strand, there is no issue regarding any presence of leftover RNA from our replication. However, for the lagging strand, there is a problem at the end because there is a primer that is remaining. That first primer you set down when starting to synthesize those Okazaki fragments, it remains. And basically, once that primer is removed, it cannot be replaced. And as it undergoes replication again, it has a shorter template than it did before. And so as DNA replication takes place, technically our genome is shortening every single time our cell divides. or genome divides. So because of that, we could end up losing important genes that are maybe necessary for survival or so forth. But what these telomeres do, specifically an enzyme known as telomerase, helps to sort of reverse that effect so that when the strand is actually cut, you're not cutting off any essential genes. It's rather just junk DNA that is placed on to minimize the issue caused during replication.